All right, so today we're going to cover cellular respiration and fermentation. Literally, this is how you get your energy. Not only is this how you get your energy, this is how most organisms get their energy, especially those eukaryotes. Cellular respiration is a cellular process of using energy from food and releasing waste products. It's all about oxidizing substrates. What is oxidation? Yes? Okay, so oxidation is when you lose electrons, right? So we're good. Everybody good on that? To oxidize a substrate means you are oxidizing it. You are taking the electron. You're taking that energetic electron, and you're going to make it do some work. We're going to also release some waste products. Now, the most popular model for cellular respiration is glucose. Glucose is easily broken down by the process of cellular respiration. You take this complex molecule and you break it apart. So in glucose, you've got, so glucose has 6 sides to it. So what kind of sugar is it? What's the prefix for 6? Hex. So it's a hexose sugar. The hexose sugar, 6 sides to it. You're going to, the first thing you do in cellular respiration is you start breaking bonds. If you break a bond, what's released? Energy. Next thing you do, you break another bond. What's released? Energy. Glucose has a ton of bonds. So it can break down things very, very easily. I'm sorry, it can break down and release energy very easily. So glucose gets taken in by the cell. Glucose gets taken in, and the cell can break it down, get some energy from it. But where does the energy in the bonds of glucose come from? The sun. So you have light shining down on what kind of organisms? Organisms with chloroplasts, like plants. Those plants are termed photoautotrophs. They're going to take the sun's energy, the kinetic energy from the sun, and convert it into the bonds of glucose. We then can use the bonds of glucose to cut them and release energy. You have 2 ways of breaking down glucose: aerobic reactions and anaerobic reactions. We're going to talk about both today. Aerobic reactions occur with oxygen. Anaerobic reactions occur without oxygen. So aerobic, anaerobic. This is the formula for aerobic cellular respiration. C6H12O6 plus 6O2 yields 6O2 CO2 plus 6H2O. Yes? Okay, so here's the deal. I'm going to ask you to not just know this, but know how each and every one of these was either broken down or built. So do you need to know it in this formula? Probably not. But you will have to tell me what happens to that glucose and that oxygen in order to create carbon dioxide and water. That's what today's whole lecture is. As we are creating carbon dioxide and water, we are also producing a whole bunch of this molecule. Katie, what is that molecule? It's not a poly thing. Take a look at it. It's got 3 phosphates attached to it. You really don't, Krissa? ATP, adenosine triphosphate. It was the one you just asked me about. That's why I wanted to give that one to you. Okay, so adenosine triphosphate. It is a major, major energy molecule in the cell. So you're going to produce a lot of adenosine triphosphate through the process of aerobic cellular respiration. There are 4 distinct stages to aerobic cellular respiration. I absolutely 100% guarantee you an essay question on your exam will be, describe the 4 stages of aerobic cellular respiration. Like, there is no doubt in my mind that is going to be a huge question on the exam. It breaks down into 4 steps: glycolysis, the breakdown of pyruvate, the citric acid cycle, and oxidative phosphorylation. Step 1, glycolysis. Step 2, breakdown of pyruvate. 3, the citric acid cycle or the Krebs cycle. And 4, oxidative phosphorylation. So we've got glycolysis, the first stage. When you think glyco, think glucose or carbohydrates. Lysis is to break down. So glycolysis is the breakdown of glucose. What's another word we've used lysis in? Hydrolysis. We're using water to break things down. So glycolysis is the breakdown of glucose. So glycolysis is not common among all living organisms, but it is pretty freaking common. If you've got access to sugar, likely you're going to have a pathway for glycolysis. Glycolysis occurs in the cytosol of the cell. It doesn't occur in any organelle. This means that even prokaryotes can undergo glycolysis. So here's what we're going to do. This is the simplified pathway of glycolysis. You start off with glucose. So glucose. This is my horrible writing. Glucose. That doesn't even show up on the screen, so it doesn't matter. You start off with glucose. That glucose is going to get a little bit of energy. Why do you have to add energy to a reaction? What's that called? Activation energy. How do you lower the activation energy? Enzymes. Every one of these steps is mediated by an enzyme. So you're going to have some energy come into the reaction. What is our primary energy carrier in the cell? Katie? No, just tell me, what's the main energy carrier in the cell? ATP. For the rest of the day, your answer is ATP, okay? So you have ATP come in. It gets oxidized and converted into ADP, adenosine diphosphate. You've released that energy. The bond was broken, energy is released. And that lets you create fructose diphosphate. That fructose diphosphate is then split in half. Okay, now you've broken a bond. What happens when you break a bond? Energy is released. In this case, energy is released in the form of NADH and ATP. So we break down 2 ATP. So we go minus 2 ATP. We gain 4 ATP. So what is our, for you business majors, what is our net total ATP for this reaction? Positive 2. 2 ATP total. And then we have 2 NADH are created. Two ATP and 2 NADH. No, not decimal values. Just total number of molecules. All right, so what we're doing then is we are taking glucose. That glucose undergoes glycolysis. In the process, net total of 2 ADP go in and 2 ATP come out. So far, so good? All right. Also, you have 2 NAD plus go in and 2 NADH come out. We're making energy here. That is all called glycolysis. That glucose, that single glucose molecule, is cut in half, though, which means you're also producing 2 molecules of what's called pyruvate, or pyruvic acid. Yes? That is a plus sign. So you're going from plus to NADH. So far, so good? Sorry, my H's, T's, I don't know what I'm drawing. Yes?
What was the whole thing about, you said that ATP go in and then 4 come out?
Yes. So that means that what is that?
Where is that?
That is right here. So if 2 go in and 4 come out, you have a net total of 2. So,
2 ADP.
so 2 adenosine diphosphate go in, net total.
Yeah.
2 adenosine triphosphate come out, net total.
Okay, wait. So, did you say there were 4 ATPs that were in here?
Okay, so take a look at what's on the slide right now, right? So you've got 2 ATP come in, 2 ADP go out. So that is 2 ATDP are made here
Oh, that has to be put in to break the fruit.
for the first step. But then at the end, you've got a total of 4 ADP being used. So you've used a net total of 2 ADP, and you've produced a net total of 2 ATP. That's all step 1. Yes?
So to describe stage 1, can you say it's the breakdown of glycolysis or glycosis, or do you have to be more specific?
I'm going to ask you to be more specific. Specifically, if you can draw this out and explain this. The biggest way to study for this part of the course is to draw pictures. Draw out these formula, draw out these pathways. Because if you can draw it out, you own it.
So would like that be an answer, like break down glucose and then that and show how it's breakdown, or?
You may want to sort of draw this down so you can explain it. Say that glucose is broken down through the process of glycolysis into 2-pyruvate. In the process, it converts 2 ADP to 2 ATP and 2 NAD plus to 2 NADH. So if you can draw it, it's much easier to explain. So this produces NADH, which is an energy transport molecule, and it produces ATP. Now it's important to note that it produces ATP using substrate level phosphorylation. What is substrate level phosphorylation? Yes? So an enzyme is directly adding a phosphate to an adenosine diphosphate. That's different from chemiosmosis or oxidative phosphorylation. With substrate level phosphorylation, it's all an enzyme grabbing and attaching. Next stage is still with glycolysis, sorry. You're going to create 2 molecules of pyruvate. You'll notice that pyruvate has 3 carbons. That's because glucose had how many carbons? 6. The glucose is a 6-carbon. Remember C6H12O6? So if you cut it in half, side note?
Yeah.
Side note, glucose has 6 carbons. Up here?
Yeah.
So sure, we can do that. How many carbons are here? 6 carbons. How many carbons are here then in each of these? 3 carbons. Because you split it in half.
Oh, yeah, that makes sense.
Make sense? This could also be a game of follow the carbon.
I got the 5.
So in summary, glycolysis is going to break down 1 molecule of glucose to produce 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of this 3-carbon pyruvate. Yes?
So would it win like that? Because on the essay, like that kind of summary statement.
Yeah, that works probably.